Oh, hello, everybody. Welcome back again. As you hopefully recall from the end of the last presentation, we are now venturing into a different area of biology. We have completed the chemistry portion of the class. So for those of you who have found chemistry a little bit of a challenge, no more chemistry in the strict sense of the word, although we will encounter a little bit of chemistry when we get to chapters 16 and 17, when we talk about the role of DNA and the synthesis of polypeptides and proteins. There's a little bit of chemistry there, but it builds on the chemistry that we've already done. So there should be nothing new coming in those two chapters. But in terms of a central focus on chemistry, we are done with that focus on chemistry. We are now working in an area that is referred to as molecular and cellular biology, rather than biochemistry, which is strictly what we were talking about in the previous set of chapters. And we begin by looking at cell division. And if you recall from chapter 1, the ability to reproduce yourself, whether you are a single-celled organism or a multi-celled organism, is incredibly important as far as the success and ongoing survival of any species is concerned. And so in terms of understanding some of the aspects of reproduction that we do need to talk about, we're going to be talking about inheritance patterns. We're going to be talking about where those inheritance patterns come from. And we're also going to be talking about the role of the DNA molecule in all of this, as I mentioned a few minutes ago. But in order to establish a context or a foundation for all of that, we need to talk about the basic process of cell division, because that's encapsulated in a very important theory that we have already mentioned all the way back in chapter 1, but we need to remind ourselves of that theory right now. And that theory is called the cell theory of life. And the first part of it is something that most people are aware of, and that's kind of obvious, that all organisms are composed of cells, whether they be multicellular or unicellular. Every organism is composed of cells, and a single cell has all the fundamental properties that we associate with life. That's why we say it is also the smallest unit of life. But the second part of the theory is what we're focusing on in this chapter and ongoing largely for the rest of the semester. It ties into this. And the second part of the theory says that all existing cells come from pre-existing cells. And so right there, encapsulated in the cell theory of life, is the notion that cells must be able to replicate themselves. So whether you are a single-celled organism where replicating that single cell is also your way of reproducing yourself as an organism, or whether they be cells within a multicellular organism like ourselves that might be replicating themselves for any number of reasons, as you'll see, the ability of cells to reproduce themselves, well, that is a fundamental property of all cells, and it's encapsulated in the cell theory of life. So we really do need to address what cell replication, cell division actually looks like. Now, if we're talking about eukaryotic organisms, the process becomes more complicated because of the presence of the nucleus and multiple DNA molecules within the nucleus. We refer to them as chromosomes, but they are at their heart DNA molecules with lots of proteins complexed with them. So we really need to address some differences between cell division as we might expect to see it in eukaryotic cells versus cell division as we might expect to see it in prokaryotic cells. And we will do that very briefly in this chapter. But what you're also going to discover is that there is more than one type of cell division associated with eukaryotic cells. And as we dig deeper into this chapter, I'll briefly touch on why that's essential, why we need to have two different types of cell division. But when we address the second type of cell division in the next presentation in chapter 13, I'll talk at greater length and greater depth about why two types of cell division are absolutely essential for eukaryotic organisms, particularly if you're going to reproduce sexually versus reproduce asexually. So more to come on that. But let's dig into this question of cell division by asking, first of all, why do we need cell division? And other than for the obvious reason, so that cells can reproduce themselves. Why do cells actually need to reproduce themselves? And there are three fundamental reasons as outlined here. So the second and the third are probably reasonably obvious. Growth and repair. I mean, it's really obvious that we do grow, and we do grow even when we reach our adult stage. When we reach our full adult form as an animal, something that is unique to animals, by the way, reaching a finite size is something that is strictly an animal feature. But even if when we reach that finite adult size, there is still some ongoing growth taking place because we're constantly reproducing or producing new blood cells. We're constantly replacing our skin. You may not be aware of this, but you turn over your skin completely every 4 weeks or so. You just do so in small fragments. You are constantly shedding small fragments of the external part of your skin, which happens to be dead cells, by the way. And they get scraped off, or they fall off as a natural part of being alive. And so as you slough off that dead layer of skin on the surface, you need to replace that. And as you don't have an infinite supply of cells to do that, you constantly have to create more cells. So there's a layer of tissue underlying your skin where cell division occurs to replace the skin cells that you are constantly losing. And then, of course, if you cut yourself, you need to heal that cut because your skin is your primary barrier to the pathogens that are on the outside desperately wanting to get in because you happen to be a good place to live. And so your primary barrier to that is your skin. So if you cut yourself, you want to heal that up as quickly as possible. And that involves cell division as well. So that's a repair mechanism. And there's ongoing repair taking place inside your body all the time. But what you may not be aware of, although if you've done a previous biology class in high school, hopefully you covered this. What you may not be aware of is that we all, as an animal, originate from a single cell, the product of the fusion of an egg and sperm, at least if you reproduce sexually. And so that united cell is called a zygote, and it is the zygote from which every cell in your body can trace its heritage. So if we go backwards, applying the idea of all existing cells come from pre-existing cells, as outlined in the cell theory of life, then we ought to be able to trace our origin and the origin of every single cell in our body back to that original zygote. And while we may not be able to do it in an organism as complicated as ourselves, with the huge number of cells that make up our body, it's estimated that there's somewhere around 10 to 13 trillion cells in your body. That's the best estimate that I've seen. Some people go higher, some people go lower, but the consensus seems to be somewhere between 10 and 13 trillion cells. But all of those cells could trace their origin back to the zygote. It would be a very complicated process to map out the origin of every single cell throughout your body back to the zygote because there are so many cells. But there are simpler eukaryotic organisms, most notably a little nematode worm called C. habitus, whose body consists of 1001 cells. And we can trace the origin of each one of those 1001 cells back to the zygote. And we can track exactly how each of those 1001 cells originated from the zygote. It's quite a remarkable piece of work to be able to do something like that. And even with something as complicated as a frog, which may not seem to be as complicated as we are, we can trace the origin in terms of groups of cells within the frog back to the zygote. And if you are intending to take the General Biology 2 class at some point in the future, if that's in your future, then we do actually get into that in one of the chapters in the General Biology 2 class. We talk about embryonic development. That's what I'm referring to here in much more detail. So if that's in your future, you'll learn a lot more about that. But for the purpose of this class and the purpose of our discussion in this chapter, all we need to remember is that in terms of our own development, in terms of our own growth, in terms of our ongoing existence, we cannot discount cell division. It is a very, very important part of our existence. So cell division, cell replication is one of those fundamental processes that all organisms are capable of. And as you'll see when we talk about the bacterial version of cell division, it's even critical there. And in fact, what you're going to see very, very soon is that there are three fundamental steps of cell division, and they are central to every type of cell division, whether it is this type of cell division that we're talking about in eukaryotic cells, whether it's the second type of cell division that we'll talk about in the next presentation, or whether it's about bacterial cell division. These three stages need to play out in sequence. And that shouldn't come as a surprise. It shouldn't come as a surprise that it's found in the two eukaryotic forms of cell division. And it most certainly shouldn't come as a surprise that it should play out in bacterial cell division, because after all, bacterial cells, prokaryotic cells, preceded eukaryotic cells. And most processes, as we have seen so far, and as we'll continue to see, that are carried out by eukaryotic cells, originated in prokaryotic cells in some form. And so cell division is really something that was invented by bacterial cells. It was then further complicated by eukaryotic cells, but that is a function of the fact that eukaryotic cells are a more complicated cell. In terms of cell division, they have a nucleus with multiple DNA molecules. So there is a level of complication that is necessary just because of that. So cell division is really central to our understanding of the existence of organisms, not just in terms of origin of cells, but in all aspects of the life of an organism. And so the type of cell division that we are referring to in this presentation, in this chapter, is referred to as mitosis. So let me write the name in here. Let me make sure the pen is active. Yes, it is. So this is the type of cell division that we are addressing in this chapter. It's referred to as mitosis, and the key point here is what this note says, that the cell division results in genetically identical daughter cells. That is true for mitosis. It's also generally true for the type of cell division that bacterial cells undergo. But as you'll see when we talk about chapter 13, that is not true for the type of cell division that takes place, that we talk about in chapter 13, what is referred to as meiosis. And for a very good reason, which we'll touch on in this presentation very, very briefly as we talk a little bit more about the basics of cell division. So here are the three fundamental steps of cell division. Remember I said there are three, and these all play out in every type of cell division, irrespective of which type of cell you're talking about. So step 1, step 2, and step 3. And the reason why I'm putting these labels on them is because at various stages, as I talk about what we refer to as the cell cycle or the life of the cell, I'll address these three stages by simply talking about step 1, step 2, step 3 of the fundamental steps. So the first fundamental step is that you have to make a copy of whatever DNA exists in the cell. Why do we need to do this? To make sure that each daughter cell gets a complete copy of the DNA. So the replication of the DNA is obviously fundamentally important here because each of the two daughter cells needs to get a complete copy of the DNA. The parent cell that is about to undergo division, before division occurs, only has a single copy unless there is a duplication of the DNA. So at some point in the life of the cell, preceding cell division, there has to be a step or a stage where we know for certain the DNA is being copied. And when we talk about the cell cycle, we can identify that, and I'll identify that for you in the cell cycle. Then once the division process begins, there is a necessity to separate the two copies. And there's a simple reason for this. This guarantees so if we were to leave the copies near each other or attached to each other in some fashion, likely what would happen is that one of the two daughter cells would get all the DNA and the other daughter cell would get nothing, or would get an incomplete copy. What we need to make sure of is that as part of the division process, each of the two daughter cells gets their own complete copy of the DNA. And one way that we can guarantee this is to simply separate the two copies. So once the two copies are completely separated, then we can divide the two daughter cells. And this is typically so one of the things that we have to bear in mind, and you'll see this very easily demonstrated in this presentation, is that the process that guarantees the separation of the two copies of the DNA usually is separate from the division of the cell itself. And this is one of the things that often gets freshman biology students into a little bit of trouble, recognizing that the division process or the separation process, the process that separates the two DNA copies, is frequently or typically separate from the process that results in the two cells of the two daughter cells, so the actual division of the parental cell into two daughter cells. So it's really important to recognize this because it becomes something of a point of confusion or a point of contention because it's so easy to think that the actual division of the cell is somehow part of the DNA separation process. And as you'll see, they are not. They are separate steps. So just to remind you again why we make a big deal out of these three steps, these are the three fundamental steps of cell division. They occur in any type of cell division that we might address, and there are three separate types of cell division that we address in this class. And it doesn't matter which type of cell we're talking about, prokaryotic or eukaryotic, we do see these three steps playing out. So these are the three steps that you need to be aware of. So the next thing is to take a look at the nature of the DNA complement in a typical eukaryotic cell, because most of our focus in this presentation is on eukaryotic cells, as it is in terms of our thinking about molecular and cellular processes. Most of our focus is going to be on eukaryotic cells, but we will address prokaryotic cells briefly where it's necessary, or where there's a lot of information to be gained by looking at prokaryotic cells versus eukaryotic cells. Here we're talking about eukaryotic cells. So in terms of the term chromosome, I have a little bit of an issue with the way the textbook uses the term chromosome, because they use the term chromosome to describe the DNA of both eukaryotic cells and prokaryotic cells. Now, traditionally, a chromosome has referred to the linear DNA molecule with its complex of protein that's found in the nucleus of a eukaryotic cell. Now, why we need to be that specific in describing a chromosome is because in a prokaryotic cell, the DNA molecule is small, it is circular, and there's a single copy of it. There's a single circular DNA molecule in all prokaryotic cells, in bacterial and archaeal cells. And they are not complex with proteins. They are pretty much naked DNA molecules, more or less. There are some proteins attached to them, but not in the way that you find proteins associated with the DNA in a eukaryotic cell. That's very different. It's very different. But I know the textbook has this tendency to use the term chromosome to reference both. If you're comfortable with that, that's fine. I prefer to reserve the term chromosome for the linear DNA molecules as found in a eukaryotic cell. That's my personal preference. So as you hear me using the term chromosome, just bear that in mind, because it may not necessarily reflect exactly what you're reading in the textbook. So with that in mind, let's take a look at how we see the DNA in a eukaryotic cell. So this is a photograph of a eukaryotic cell that is in the process of dividing. And the reason why you can tell that it's in the process of dividing is simply because you can see the chromosomes. So we can say here normally because they're in their chromatin form, and we've talked about this before. Oops. The chromosomes become visible as division begins. And the process is actually referred to as condensation, or we say the chromosomes are condensed and become visible. So these chromosomes are stained with a stain that highlights them so that you can see them a little bit more clearly, but even in their unstained form, they're quite visible in a light microscope. A little bit tougher to see, but you can still see them. excuse me, by virtue of the fact that they are condensing. So they haven't completed their condensation, the condensation process yet. So they're in the process of condensing. And what this does is fold the DNA up. We've talked about the notion of coiling when I introduced you to DNA back in chapter 5. So the DNA here is coiling and supercoiling, which is a convenient way of packaging it. It shrinks the length of the molecule down, it shortens it and thickens it, and then it gets coated with a layer of protein to protect it, because as you'll discover, as we talk about the stages of mitosis, at one point in mitosis, the nuclear membrane actually breaks down, the nuclear envelope breaks down, and the chromosomes become exposed to the cytoplasm, which is a very hostile environment for DNA. So the way that you can protect the DNA during this short period of time that the chromosomes are exposed to the cytoplasm is to coat them with protein to act as a protective layer. The added benefit of this condensation is that it makes it a lot easier to move the DNA around rather than in its chromatin form. It's kind of the equivalent of trying to move a piece of string, a long piece, say a 10-foot piece of string, as a long 10-foot piece of string versus rolling it up into a little ball and carrying it. It's obviously a lot easier to carry the little ball of string than it is to carry the 10-foot piece of string. And so it's basically the same notion here. It's a lot easier to move the chromosomes around, and they do get moved around during the stages of mitosis, the stages of cell division. So it's a lot easier to move them around in their condensed form than it would be if they were in their chromatin form. And bear in mind that prior to this division process beginning, the chromosomes actually get duplicated. So we're talking about not just the original chromosomes as they existed in the cell, we're talking about 2 copies of the chromosomes. That makes it even more essential to have them in a convenient way to move around, and condensing them is obviously a good way to make it convenient to move them around. But this happens right as the cell begins the division process. So if you're ever wondering, how can you tell if a cell is about to begin division? Take a look at the nucleus. If you can see that the chromosomes are starting to become visible rather than just an amorphous appearance of the nucleus, you actually start to see the chromosomes as visible structures inside the nucleus, then you can be very confident that the cell is starting the process of cell division. That's a big giveaway. And it is in fact one of those important observations that everyone learns about how to recognize a cell beginning the process of division. It's one of the big giveaways for recognizing the first stage of cell division, as you'll see. So when we start thinking in terms of the 2 different types of cells, basic type of cells that you're going to find in an organism, the general cells, what are referred to as general body cells or somatic cells, have a complete complement of all the chromosomes. And we say that they have 2 complete sets of chromosomes. So you can see here the term 2 complete sets of chromosomes. Maybe I should put that in red. Erase this and put it in red. So 2 sets of chromosomes. And we have a term for that. And you'll learn this again in chapter 13, but I'll give it to you here because it's a good way. Oops, let's make that neater. You can do better than that. The term here is, even that's not good enough. I want you to be able to read this. Let's try this one more time. There we go, diploid. So somatic cells have two complete sets of chromosomes. We refer to those as diploid cells. Gametes have one set of chromosomes, and we refer to that as haploid. So it's not hard to imagine where these sets of chromosomes may have come from. So for humans, we say that somatic cells have 46 chromosomes, so the diploid number of chromosomes for a human is 46. And gametes, the reproductive cells or sex cells, and perhaps I should put a term, these would be the eggs and sperm. They are what are referred to as gametes. They have 23 chromosomes. And so immediately you see gametes or reproductive cells are not identical to somatic cells. And given that somatic cells, when they make copies of each other, are going to make sure that they're genetically identical to each other, it stands to reason that whatever process of cell division somatic cells are using would not be appropriate for the production of gametes. Hence the requirement for a second type of cell division, which is, of course, the subject of Chapter 13. So in thinking about the sets of chromosomes, given that this is connected to sexual reproduction, I mean, eggs and sperm, that's sexual reproduction, it stands to reason then that if we think about the sets of chromosomes that are present in our cells, one set of chromosomes must have come from one parent, one set of chromosomes must have come from the other parent. And so when we start talking about the other type of cell division in Chapter 13, which is referred to as meiosis, one of the things that we need to bear in mind is that that type of cell division has got to create a situation where we can give rise to eggs and sperm, to gametes that only have one set of chromosomes rather than the two sets of chromosomes that are going to be present in a somatic cell as a result of mitosis. So more to come when we talk about Chapter 13 next week. So if we remind ourselves again of the three fundamental steps of cell division, remember the first fundamental step involves duplicating the chromosomes. So at some point in the lead up to cell division, there's got to be a duplication of the chromosomes. And initially, those duplicated chromosomes, which we refer to in eukaryotic cells as sister chromatids, are going to be anchored together by a protein complex. And so when you take a look at these duplicated chromosomes during the process of cell division, this is what you're going to see. You see the sister chromatids. So one copy of the DNA molecule, one copy of the chromosome is sitting there. The other copy of the chromosome, oops, let's get rid of that, is sitting there. And they're anchored together by a protein complex that's sitting here, referred to as the centromere. So these two sister chromatids make up a chromosome. But there's a problem with this that is illustrated in the next diagram. So let me take you to the next diagram. And here's the problem. This structure consisting of a single DNA molecule, which technically speaking never exists in a cell prior to cell division. After the cell's complete cell division, the chromosomes decondense back to the chromatin form. So you're never going to see a chromosome in a cell that has this form. That the artist has taken a little bit of liberty here. But this single DNA molecule, as it would exist in the cell, this is a chromosome. We've already said that this, with two DNA molecules in there in the form of the two sister chromatids, well, this is also a chromosome. And unfortunately, when the two sister chromatids separate, which they must do during the cell division process, remember step number 2 says that there has to be a separation of the duplicated DNA. Well, the sister chromatids represent the duplicated DNA, so at some point they've got to be separated. We refer to each of these as chromosomes. And this is something that biologists and scientists in general are pretty good at, utilizing the same term in multiple ways. And so here we have 3 different ways of referring to a chromosome. And so to separate out this unique form of the chromosome that has the 2 sister chromatids representing the 2 copies of the original chromosome, I and most biologists refer to these as duplicated chromosomes. And as we talk about mitosis and cell division in general, I'll try and remember to refer to this structure as a duplicated chromosome to separate it from, or to differentiate it from, chromosomes that consist of single DNA molecules. So where you've got a chromosome that's composed of 2 sister chromatids representing the duplicated chromosome, I'll refer to this as a duplicated chromosome. Whereas if we have a single DNA molecule present in the chromosome, then I'll just refer to that as a chromosome, so that we can make a distinction as we go through. And remember, again, this is something that can get really confusing. In fact, one of the things that I tell all of my students is that if you can wrap your head around this diagram and understand the difference between a single chromosome and a duplicated chromosome and what happens to the chromosomes during cell division, you can go a long way to understanding mitosis and meiosis by simply making sense of this diagram and understanding the connection between a single chromosome or a single DNA molecule as a chromosome, this structure as a duplicated chromosome containing 2 sister chromatids, 2 DNA molecules, and when they separate, the fact that each of these now becomes a chromosome in its own right. If you can make sense of that, if you can just wrap your head around this diagram, it will make mitosis and meiosis so much easier to understand. So this is kind of an important diagram to focus on. So we come to our first break. We've been going for a little while, about 30 minutes, I think. So this is a good place to take a break. And so here is my concept check question. Remember, I am going to turn off the recording at this point, having stated what the concept check question is. And when we come back to the beginning of the next recorded section, I'll give you the answer to this concept check. So irrespective of whether you're just going to click through and follow along with the ongoing recording, whether you're actually going to physically walk away or stop for a few minutes and then come back again, you'll get the answer to this when you start up watching the recording again. So here's my concept check question. A chicken, like all birds, has 78 chromosomes in its somatic cells. How many of these chromosomes came from each parent? How many chromosomes would be in the chicken's gametes? And how many chromosomes would there be in the somatic cells of an offspring of this chicken? I'll see you on the other side of the break.